Greetings all, my name is Khanyisile Mngomezulu. I am a PhD candidate from the University of KwaZulu-Natal. My talk for today is entitled An In Vitro Investigation of a Traditional Medicine Product with HIV Therapeutic and Curative Potential. So although combination antiretroviral therapy can successfully suppress plasma virus levels to below the limit of detection, it is not curative. And this is because viremia is still able to rebound rapidly after treatment interruption. And this is due to the early formation of stable and long-lived latent reservoirs. A latent HIV infection is established when the HIV virus infects activated CD4 T cells, and this leads to viral replication and later on cell death due to immune responses. However, a fraction of these cells may become resting memory T cells and form part of the latent reservoir. Furthermore, a less frequent alternative mechanism for latency is the infection of resting naive CD4 T cells. So the latently infected resting naive or memory CD4 T cells may become activated, leading to viral replication and later on cell death. Therefore, the latent reservoir is a major drawback to finding a cure for HIV. So different cure strategies have been developed in order to eradicate these reservoirs, such as the shock and kill strategy and the block and lock strategy. So the shock and kill strategy involves forcing viral gene expression by the use of latency reversing agents while maintaining CART in order to avoid new rounds of infection. So the now infected cells are then killed via targeted immune responses. In contrast to the shock and kill strategy, we have the block and lock strategy that uses latency promoting agents that block HIV transcription and reactivation in latently infected cells in order to achieve HIV remission. So for the purposes of this study, we are focusing on the shock and kill strategy. So this was a study that was done by Bullen et al. In 2014, where they investigated the potential of different latency reversing agents in their ability to reactivate the latent virus. But what we are seeing here is that these latency reversing agents were not potent in reactivating the virus when compared to the positive control, which was ionomycin and PMA, meaning that there is still more research that is needed to be done in order to find more LRAs that are potent, not toxic, and inexpensive. So as a new alternative, we introduced traditional medicine. So approximately 66.7% of the population in KZN in South Africa consults traditional healers for the management of their diseases. And this includes using traditional medicine to treat HIV infection. Furthermore, natural products from plants and marine organisms are a promising but undervalued resource for identifying new antivirals. So the study hypothesizes that a traditional medicine product termed Product Nkabinde will reverse HIV latency by inducing pro-virus expression across multiple cell lines. And the study aims to investigate a plant-based traditional medicine as a potential agent to reactivate resting CD4 T cells and induce transcription of latently infected HIV. So in order to achieve our aim, our objectives were to evaluate the cytotoxicity of the traditional medicine product on an in vitro lymphocyte cell line model. Secondly, to evaluate the antiviral activity of the traditional medicine product in HIV subtype B and HIV subtype C infection. Thirdly, we wanted to determine the reactivating effects of the traditional medicine product in JLET's latent HIV model cell line. So the traditional medicine that we are using was obtained from Mr. Nkabinde, and the medicine is a combination of 4 plants, which we termed the mixture, and we are also using a single plant, which we termed SDK2. So the medicine was prepared by the healer at his home, and in the lab, we wanted to determine the safety profile of the medicine, and we did this by conducting cell viability tests where we treated cells with different concentrations of the medicine in order to find the half maximal inhibitory concentration. And this was done using the cell titer glow assay, which measures the number of viable cells. So this was measured using the luminometer. So when we had obtained our IC50, we used that IC50 to treat TZMBL cells. After treatment of TZMBL cells, we then infected with 2 subtypes subtype C viruses and 2 subtype B viruses. After that, we then went on to now assess for virus inhibition using the luciferase assay. So this was the results that we obtained after conducting the cell viability test. So these are the concentrations that we obtained and we are currently using for treatment. So these are the results we obtained when we wanted to determine whether this medicine does have antiviral activity when compared to the positive control efavirenz in figure A. And what we found was that both the crude extract, the mixture in figure B, and SDK2 in figure C showed a dose-dependent inhibition for both subtype B and subtype C viruses, paying closer or special attention to the mixture, which was more potent in inhibiting these viruses when compared to the one plant, SDK2. So we then went on to investigate whether the medicine does have latency reversal potential. So we then used the JLET model where we stimulated the JLET subtype B and JLET subtype C cells with concentrations of the mixture, SDK, and other LRAs for 24 hours, and then we assessed for GFP. So these are the results that we obtained when we stimulated the JLET subtype B and JLET subtype C cells with the medicine. So when we stimulated the JLET subtype B cells with 325 micrograms per ml of the mixture, we saw 1% GFP positive cells. And when we stimulated the JLET subtype C cells with the same concentration, which was 325 micrograms per ml, we saw 0.8%. As we can see, the mixture showed very low activity when compared to PMA, which was the positive control. Interestingly, when we stimulated the JLET subtype C and subtype B with 106 micrograms per ml of SDK2, we found 25.3% GFP positive cells for the JLET B cells and 14.9% for the JLET subtype C cells. So these results showed that the one plant did show some reactivation potential. So after seeing that the medicine does show reactivation potential, we then went on to now combine the medicine with different latency reversing agents. So the latency reversing agents that we investigated was penobenostat, SAHA, and TNF alpha. And from this data, we can see that these latency reversing agents alone stimulated the JLET subtype B cells, and we saw no activity for the JLET subtype C cells. And when in combination with the mixture, we saw the same activity as shown in the controls, so it was more or less the same. However, when we stimulated with SDK2 in combination with the LRAs, we saw higher percentages of GFP positive cells for the subtype B, and interestingly, we saw reactivation for the JLET subtype C cells. So in summary, we were able to show the 50% cytotoxicity concentration for SDK to be 106 micrograms per mol and 325 micrograms per mol for the mixture. Secondly, the SDK and the mixture showed potent antiviral activity when compared to efavirenz, which was our positive control. Thirdly, we were able to show that the single plant showed potent reactivation potential when compared to the mixture, which showed low activity. Lastly, SDK was able to enhance the activities of the individual latency reversing agents in JLET subtype C cells. So lastly, I would like to acknowledge the following individuals, institutions, and funders. Thank you.